In this video, we will discuss about how this cytochrome C, when it is present in the cytosol, can trigger apoptosis. The natural location of the cytochrome C is within the mitochondria, where it helps in the ATP formations. As we know that mitochondria is known as the powerhouse of the cells. And so the cytochrome C helps in the ATP formations within the mitochondria. And when it is somehow leaks out into the cytosol, it will interact with this APF1, and that will help in the apoptosis formation. So apoptosis is mainly triggered within the cells through two different pathways. First one is the extrinsic pathway, another one is the intrinsic pathways. Though we have already covered the extrinsic and intrinsic pathway in one of our previous videos, we will quickly revise those things so that we will understand how this cytochrome C uh, plays a role in the apoptosis. In the case of extrinsic pathways, the DAT ligands will bind to the DAT receptors that are present on the extracellular matrix, and that will trigger the downstream signaling pathways, where it will lead to the formation of pro-caspase 8 or caspase 8. And this pro-caspase 8 or caspase 8 can then interact with this can later interact with this cytochrome C, can then interact with the cytochrome C that is shown here. The cytochrome C, two things happens when this pro-caspase and caspase formation occurs. First one is that this inactive BID will convert it into the active BID through truncation of this BID to this TBID, and this TBID then go inside the cells, and thereby it will force the cytochrome C to leaks into the cytosol, where this cytochrome C interacts with this APF1, and this complex can cleave the pro-caspase 8, that will lead to the formation of this caspase 3 and caspase 7, and then this caspase 3 and caspase 7 can trigger apoptosis. So in the case of other apoptotic pathways, it will trigger these BCL2 families. In this BCL2 families will cause the activation of this BID through truncation of this BID, and thereby that will lead to the release of the cytochrome C into the cytosol, and then further process will remain similar. That is, it will cleave this pro-caspase 9 to form this caspase 3 and caspase 7 that will trigger apoptosis. This is how the extrinsic and intrinsic pathways triggers apoptosis. Now let us understand how the normal conditions behave. In normal conditions, the DAT receptors does not get interact with the DAT ligands, and the cytochrome C remains within the mitochondria, and also the component that is similar to the cytochrome C is the SMAC. It is also remain within the mitochondria, and in this case the APF1, though it is remain in the cytosol, it remains inactive and it will not cleave this pro-caspase 3, 9, 7, and thereby there is no triggering of apoptosis occurs. And in this case also, this BCL2 and BCL XL will act as anti-apoptotic factors that will prevent the leakage of cytochrome C into the cytosol. So in the how this leakage of cytochrome C into the cytosol occurs is whenever there is a stimulus of apoptotic, either through extrinsic or intrinsic, and some of the factors could be the DNA damage or unfolded protein response. It will lead to the cleavage of this BID that will lead to the formation of truncation BID, and it will lead to the oligomerization of these bugs that in normal case remain separated apart, and this oligomerization of the bugs will lead to the pore formation, and that will cause cytochrome C release into the cytosol, and then it will act the way that has been shown here on the figure on the left hand side. The other ways by which cytochrome C can be leaked into the cytosol is through this PTP pathway, where because of the differentiation, because of the difference in the amount of Ca2 that can be in the cytosols and also in the mitochondria, can cause the pore formation that will lead to the release of the cytosols. This will lead to the release of the cytochrome C into the cytosols, and this could happen because of the ER stress in the ER stress could be because unfolded protein response, or due to disbalance of the calcium cycles that is maintained by the ER stress. And we have covered the ER stress in our several videos. You can go through those videos to understand how this ER stress is developed and what are the signaling pathways that get activated. And this SMAC. So now let us understand how this cytochrome C can be inactivated, or what is the feedback loop. Cytochrome C can be inactivated by these HSP27 proteins and also these IAP proteins. So this will cause the inactivation of IAP, cause the inactivation of caspase 9, caspase 3, caspase 7. Thus it will prevent the apoptosis, and the HSP27 prevent the leakage of cytochrome C into the cytosol. Thus it will prevent the activation of APF1, and thereby the apoptosis will get inhibited. However, this SMAC will cause the inactivation of this IAP. Thus, it will support. The apoptotic conditions, and thereby the SMAC is considered as. Thereby, the SMAC in this case is considered as pro-apoptotic factors, and this IAP and HSP27 is considered as anti-apoptotic factors. And these things that where it is this caspase 9 oligomerization interacts with the cytochrome C is the active pro-apoptotic factor, since it will lead to the cleavage. It will lead to the formation of this caspase 3 and caspase 9 that will trigger apoptosis. So in this case, there are two apoptotic factors, HSP27 and IAP, and this oligomerization of these bugs leads to the leakage of cytochrome C into the cytosol that will cause the apoptotic triggering. So I hope you got the idea how this how this cytochrome C into the cytosol can lead to the how cytochrome C into the cytosol can trigger apoptosis, and what are the anti-apoptotic factors that prevents the leakage of cytochrome C to cytosols, and what are the factors by which cytochrome C can be triggered. If you need to understand about this thing in detail, you can go through the reference that has been provided in the description.